Hey everybody, Mr. Murphy here. This is 10.3, which is all about codons and mutations. I apologize if you're watching these in order because in the last video I said this was going to be about translation. That comes later. So, when we look at the genetic code, it is a series of sets of three, which we call codons, which specify which amino acid should be added on to a growing protein. So when we take DNA and then we turn it into RNA, that RNA, we look at these triplets called codons. There are start codons, which initiate the process of translation, and stop codons, which tell the ribosome when to stop. In terms of experiments to discover this, it's relatively simple in terms of looking at different regions of an RNA and whether those have all U's or all A's or all C's. We can demonstrate first off that we get three products and that those three products are the same within the single strand. So I get three phenylalanines here and I get three lysines here and three prolines here. And so that demonstrates that it is sets of three that matter, and also that what is adjacent to those has no effect. So this set of three is not affected in any way by whatever comes next. So all of this is kind of stuff we just take for granted at this point because you've already been taught it before. Here is the codon chart, and just talking a little bit about this, it's important to know right off the bat two things. The genetic code is very redundant, and it's also not ambiguous. And what that means is that each codon codes for only one amino acid. It's not ambiguous. It is very specific, coding for one amino acid. But for any of these amino acids, you notice that there are typically multiple different combinations of codons that can cause that amino acid. And we'll see how that actually is quite a benefit when we consider mutations that can occur. And simply put, the fact that this U here has changed to a C had no effect on the successful selection of the amino acid and therefore the successful creation of the protein that was desired. There are some exceptions in terms of how this codon thing works within organisms, but what's really cool is that this is pretty much universal. And what that means is that every organism on the planet, again, with minor exceptions, and those exceptions are just slight tweaks, they're not fundamental differences, but nearly every organism utilizes the same triplets to code for the same amino acid. And that's not required. There's nothing that says that glutamine can only be associated with these three codons. When we look a little closer at the tRNA molecule, you'll see that the tRNA just has what's called an anticodon that is the opposite, the complementary bases of these. It's one tiny little region. It has nothing to do with the amino acid that's attached to it. Now, why does that matter? Well, it matters because that shows us how genetics and DNA and RNA are the common thing between all forms of life, which has huge implications for the concept of evolution and the common ancestor of all living organisms, and how DNA in this genetic code dates back all the way to the beginning of life on Earth. The fact that you look at any different organism, even widely different ones, bacteria or people or plants or fungi, whatever, they all utilize the same exact process of conveying that genetic information between generations, which, as we'll talk about more in our evolution unit, really couldn't have independently arisen. Because like I just said, there's nothing that says it has to be like this. It could be completely reorganized or done in an entirely different mechanism altogether. So, pretty cool. Mutations, not cool, usually. Mutations are simply put changes in the genetic code. And mutations can occur at any point, but typically the mutations that we care about are mutations that occur to the DNA, because those are going to be persistent. If you have an RNA that gets created with a mistaken single nucleotide, it's probably not going to be the end of the world, because that single RNA is only going to account for a very small number of products, whereas the next time we transcribe it, that mistake probably wouldn't get made. So it's mistakes in the DNA in terms of mutations that are most worrisome, because those are going to persist through the entire life of that particular cell. Or mutations that occur in the very early stages of development, or in the DNA that's contained inside of the egg or sperm, those are going to be persistent, sort of more widespread throughout the organism. So again, mutations and where and how and when they occur all play into sort of what the severity of the effects of that will be. Now, silent mutations are the first type to talk about, and that's the ones that you wouldn't ever know that they had even occurred. And what I mean by that is that a silent mutation leads to a switch of a nucleotide to one that still codes for the same amino acid. And so even though this has become an A instead of a C, it still codes for the same, I guess this would be the proline here, the same amino acid. And so the product protein, you would have no way of knowing that that had happened. Now, silent mutations are not completely unimportant, however, because this change from C to A, yes, right now, leads to the same product. But it has now set the stage for a second mutation, were one to occur, to lead to a fundamentally different change. You know, if this G changed to something else, who knows? This may be an entirely different amino acid because now we have an A here instead of a C. So silent mutations, in an additive sense, can lead to pretty significant differences. And so looking between different organisms and the silent mutations that account for some of the genetic differences in common proteins between, let's say, like a person and a cow, those silent mutations can have more pronounced effects down the road, or combined with other mutations and other genetic stuff that's going on. Missense mutations, however, these are substitutions where a single nucleotide changes and it causes a single nucleotide to be different. Now, this may be not a big deal. Some proteins are made out of hundreds or more amino acids, and so that maybe one, got a proline instead of a leucine. Maybe that's not a big deal, but it can be. And missense mutations can result in defective proteins and reduce the protein efficiency. There's also examples like the Tp53 gene where a missense mutation causes a gain of function. And that sounds like, oh, cool, so you can like see through walls or fly or something, right? No, sadly, the Tp53 gene is associated with cancers, and I think ovarian cancer in particular, where the Tp53 mutation is very prevalent. And what it causes is the P53 protein, which is a tumor suppressor protein, kind of gains this ability where it begins to promote cell division, where ordinarily it's more part of the monitoring system of cell division. And so it has gained a unfortunate function as a promoter of cell division. So missense mutations, while they are small and may not seem like much, they can have pretty profound differences. We'll also see other examples of like a single missense mutation which could lead to an entire extra body segment on insects. So they can be quite profound if they happen in just the right place or just the wrong place. Depends on how you look at it. So here's a missense mutation where this T became an A, which changes then the, instead of an A here, we have a U in the RNA, which then codes for a different amino acid. Nonsense mutations typically are quite problematic because a nonsense mutation causes a substitution, a change of a nucleotide, which now prematurely codes for a stop codon. So you might be halfway through building a protein with your ribosome and then it hits a stop codon that shouldn't have been there. And in that case, then you typically are going to be getting proteins that are pretty fundamentally broken and unable to have whatever their intended effect is. So here we go. Here's a whole segment of DNA that should be coding for a whole big long section of RNA, except, whoops, our second amino acid happens to be a stop codon. So whatever that protein was, it's certainly not doing its job in this case. Frameshift mutations are a little more complicated, and this is where we actually get an insertion or a deletion of bases in the DNA. So instead of just having, this became something else, now between this C and this G, there's a added thymine. So it's sort of snuck in there. And so in this case, the reason why this is such a big deal is because we do deal with everything in threes, except now we put in an extra letter. And during transcription and translation, there's no mechanism to say like, oh, wait, that's an extra letter, don't count it. And so instead now we're looking at an entirely new series of triplets. And so after this point, instead of being, TAC, ACC, GAG, GGC, that T now has shifted the entire set of of codons, and so now we have this codon is different, and this one is different, and this one is different, and this one is different. And so again, can be very detrimental to the intended effect of that protein, because now every single codon after that point is going to be wrong. And so it's similar to the nonsense mutation in that it causes, you know, a completely screwed up protein, but different because it still can continue coding for amino acids and make some wildly variable product, which could be, you know, best case non-functional, worst case somehow damaging. And that is a little bit about mutations, and next up will be translation. Let me know if you have questions. Thanks.